Justin Santopietro with the MAHA Institute. Thank you for traversing the frozen tundra to join us here today. This is a very exciting event. I think just the fact that this event is happening is testament to how different this NIH leadership is to its predecessors, and we're so grateful to have you all here today for what I'm sure is going to be a really great conversation. So I'll go ahead and do some introductions here. Since Jay was already introduced, I'll start with Dr. Matthew Memoli. Dr. Memoli is the Principal Deputy Director of the NIH. He briefly served as Acting Director from January 22nd to March 31st of last year. He's worked at NIH for more than 20 years, most recently serving as Director of the Clinical Studies Unit within the Laboratory of Infectious Diseases, part of NIH's NIAID. He previously served as a staff clinician in the same laboratory, and he's an internationally known expert in the field of respiratory viruses. He's won multiple NIH awards for his work and recently served on the Governor of Virginia's Medical Advisory Board from 2022 through 24. Dr. Memoli is a graduate of William & Mary and received his Master of Science in Microbiology from Thomas Jefferson University. He completed medical school at St. George's University School of Medicine and his residency at Georgetown. Dr. Memoli joined NIH in 2005 to complete his fellowship in infectious diseases. Dr. Nicole Kleinstreuer on the end. She is the Deputy Director, I'm going to take a big deep breath here, for Program Coordination, Planning, and Strategic Initiatives, DPCIPSI.
There's too many acronyms in the NIH. I don't think I could fix that, though. It's just too rooted in the culture.
In this role, she leads the division within the Office of the NIH Director, which oversees trans-NIH programmatic research and strategic policy initiatives. Dr. Kleinstreuer is internationally recognized for her leadership in developing innovative, human-relevant research strategies that advance public health protection. In her prior role, she led an interagency and international efforts to promote new approach methodologies, reduce animal testing, and integrate computational modeling, AI, and systems toxicology into regulatory science. Her work spans translational bioinformatics, predictive modeling, and quantitative risk assessment. She's the author of over 150 peer-reviewed publications and has received numerous honors. She got her BS in biomedical engineering from UNC Chapel Hill and PhD in bioengineering from the University of Canterbury. She is deeply committed to mentorship, public health protection, and scientific innovation that enhances the translational and biomedical research for real-world impacts. And finally, Dr. Anthony Letai is the director of the National Cancer Institute. He joined NCI as the 18th director in September of last year and previously served as professor of medicine at Harvard Medical School and medical oncologist at Dana-Farber Cancer Institute. Dr. Letai is a physician scientist whose work has helped transform cancer treatment and advance functional precision medicine. He received his Bachelor's of Arts in Physics from Princeton, his MD and PhD from the University of Chicago, and completed his residence in internal medicine at Brigham and Women's Hospital and a fellowship in hematology and oncology at the Dana-Farber Institute. So as you can see, we have an incredible lineup here today. So thank you all again for joining us. So Dr. Bhattacharya, you already sort of started with this panel is called NIH Then, and now you sort of touched on the then part. But I do want to go back to that period of time a little bit briefly. During the COVID era, many Americans came under the impression that Anthony Fauci was the NIH director because we saw him on TV every day for weeks on end. Right?
Some people still think that.
Yeah. Some people probably still think that. When in fact he was in charge of just one of the 27 institutes and centers. Which is to say, there's a lot more to NIH than most Americans probably realize. So to start us off here, can you give us a sense of NIH's size, its structure, and its role in the federal government and biomedical research world?
Sure. So the NIH is the single largest public funder of biomedicine in the world. I think like 85% of public biomedical funding, just taking all, you include foundations and you include all the other countries in the world, 85% of public funding for biomedicine comes from the NIH. It's about $50 billion a year, which is, it's a lot of money, right? And yet there's estimates that for every dollar invested in the NIH, it produces $2 to $5 of like economic benefit and its mission, the fundamental mission, as I mentioned earlier, is to support research that improves the health and longevity of people. There's 27 institutes and centers. Before I became director, I sat down and memorized all of the acronyms for every single one of them. It's a daunting task. But the thing about that task is that if you look through what the activities are, nearly every single area of biomedicine the NIH touches. And in fact, not just the NIH touches, it leads. Almost every single biomedical researcher of note, not just in the United States, but almost worldwide, have had NIH funding or support or training or collaboration in some way. If you have gone to your doctor, if you've gone to your chiropractor, if you've gone to if you've ever been taken care of by a nurse, if you've ever gone to a pharmacy, the NIH has been part of that experience. Tony Letai leads the National Cancer Institute. Almost every single cancer medicine, every single cancer therapy, every single advance in cancer that has happened in the last 50, 100 years, the NIH has been sort of at the root of. So it's way more than Tony Fauci. We're going to have the anti-Tony Fauci in a second, and in the next panel coming up. But but also in infectious disease. Infectious disease is the conquest of infectious disease, including the development of antimicrobial agents for bacterial infections, research into more effective ways of preventing infectious disease, treatments. At the heart of it is NIH investments. Immunology, the fundamental knowledge we have in immunology, part of which informed my thinking during the pandemic in fundamental ways, came out of, again, NIH investments. Nearly every single area of biomedicine that is of importance to people has been funded or supported or undergirded by Americans funding the NIH.
Got it. So this crew, you guys have been at it for less than a year, but you've already engaged in some of the most far-reaching reforms that NIH has seen in many decades. So what were some of the, if you can indulge us, what were some of the worst problems you encountered when you first took the helm about a year ago? Yeah, this is for everyone.
He was the director before I was, so that's just, well,
That happens.
So, so
you should tell the story.
well, yeah, okay. So he's telling me to tell the story how I became director. Okay, you mean the actual moment it happened? Is that what you're talking about? So I was so as you heard earlier, I've been working at NIH in the intramural program. Jay didn't talk about this. So NIH is broken up into sort of two parts: the intramural program and the extramural program. The extramural program is all that funding that Jay was talking about that we send outside of NIH's walls to universities and companies and all the different people that we fund. The intramural program is much smaller, approximately about 10% of our budget, that goes towards scientists working on the NIH campus as academic researchers. And those are also the folks that run studies in the clinical center, of which I have been one for about 20 years. And so I was working in my lab, and I got a phone call from the White House liaison saying, "You're the NIH director. Go to Building 1." And I said, "I think if I go to Building 1 and tell them I'm the NIH director, I'm going to be arrested." So I said, "I don't think that's going to work. Have you told them yet?" And they said, "No." And I said, "Well, maybe you should tell them before I go over there." So it took another 2 days. And then they called me back up and said, "Okay, they know you're coming." And then I walked in, and it was a big shock to everybody, I think. But getting to the question, I think that the biggest problem that I faced when I walked in the door was that we really hadn't had significant leadership change at NIH for at least 17 years, and you could probably argue even longer with some of the other leadership that we had. And there was some, I think very specific ways that things had been done and ways of thinking around the institution that had really become sort of a force within NIH, and some of that was not good. And so coming in, one of the first things I did was really, sort of push against certain things to say, hey, we need to do things a little differently. We need to start thinking about things a little differently. We need to move faster. We need to, take more risks. And there was a lot of resistance to that. And I think slowly, especially over the past year, we've made enormous strides in breaking through that resistance. And so I guess if I had to put a set of words to the biggest problem was institutional aversion to risk or aversion to institutional risk. They were worried about change, worried about taking chances, worried about, what's the scientific community going to say? What's the media going to say? And that's not a way to make progress, right? You have to take chances and do things. And if you find out you're wrong, you admit you're wrong and you change course. And I think that's how we're looking at this. We're making big changes. We're moving quickly, but we're evaluating every step of the way. And if something's not working, we'll change and do better.
One of the most fun things about being an NIH director has been to find people that come to you and then say, well, I've had this idea for 15 years. I mean, this is Matt can attest to this. Actually, Nicole is an example of this. I met Nicole, and she told me she'll tell you about the ideas she told me in a second. They say, look, I've been inside the NIH for a decade, and I can't get any progress on this. And you just look at them and say, well, that's a good idea. Let's do this. And they just look in shock. They're like, what? Wait, we could just do this? This happened time after time after time where people, really brilliant people, the scientists of the NIH come to you and they just give you these ideas. And they're like, well, why haven't you done this? And really, fundamentally, it's the stasis that Matt's talking about.
I'll just add to that. So that in addition to that stagnation, I think that something else that we've all observed is this kind of fragmentation. So having each of the institutes and centers, although they meet at a minimum once a week and talk to each other, they still have or had relatively isolated portfolios. They had their silos, they had their areas where they were funding, and the DPCPC, one of those fun government acronyms, and the division that I lead is Program Coordination, Planning, and Strategic Initiatives. And the responsibility of DPCPC, so there's 15 different offices under me that coordinate cross-cutting research themes across the NIH and the larger scientific enterprise. And I was shocked to learn that the vast majority of people that I came across at NIH had never even heard of DPCPC. Well, that's a huge problem. If you have a centralized function whose role is to actually coordinate across all of the different institutes and centers and no one's ever heard of it, then it's not really doing its job effectively, right? And so one thing that's been incredibly refreshing under Jay and Matt's leadership is to see this sort of forced communication and really strong encouragement for the different institutes and centers to come together and to work as one NIH rather than working as 27 individual institutes. And there's so much more progress that can be made and accelerated when you have the NIH working together as a whole, because as you all well know, things like the chronic disease crisis require a multi-pronged strategy to address.
I'd just like to comment, I think the NCI, in some ways, lives a blessed life. I think most of you, if you reflect on some of the troubled years of the COVID infections and some of the loss of trust in public health, to some extent, the National Cancer Institute and the sort of National Cancer Program more generally stayed out of it. I think one of the nice things when I've been told that being National Cancer Institute director is like the best job in government. And that's because it's a rare place where most people are your friends, actually, from both parties, from all levels of government. They want you to succeed. And I didn't really come to of course I have ideas to change things, and we can talk about that later. But as far as sort of like real reform, I think most people felt that the NCI had been largely doing its job. And I think that actually can be reflected in some of the successes that we've seen over the years, like a dramatic reduction in cancer mortality, a reduction of 30% since 1990. That doesn't mean I don't want to change things, but I think the NCI is very fortunate in that it wasn't particularly controversial, didn't sort of embrace a lot of the problematic ideas that I think may have been present elsewhere. And I think before taking this job, I had the privilege of meeting not only with Jay, but also with the secretary and the president. And I wasn't given a project of reform. In 2 words, they sort of told me to cure cancer. And I think we can all agree. I think we can all agree on that, and that is exactly what I'm going to try to do.
Thanks. Before we go on, I should mention, if you have questions for any of our panelists, you can write them down on the notepads on your table, and one of our team members will collect them, and we'll try to get through those in the last 15 minutes of this session here. So, under the task you have to turn this massive ship of the NIH, which sounds like it's like 27 different ships that are sort of tied together, is a incredible undertaking. You've faced enormous amount of backlash. Anytime you try to change extremely entrenched processes, it's going to be difficult. And a lot of the coverage in the press and the political angle has focused on the negative stuff, the complaints and dissent from within and without. But we all know, everyone who works in the scientific enterprise in the United States and beyond knows there are deep-seated problems in how this works, in the incentives, in the structure, and that they need to be addressed, and they've just been kicked down the road over and over again. So I'm really curious to hear from all of you some of the positive comments you've gotten from your colleagues at NIH or out in the research world about now that things have been cracked open a little bit and there's really an opportunity for change. I'd really like to hear about the positive stuff you guys have been hearing.
Let me give one. Okay, so I got to set this up for a second, right? So the way that sorry, I'm a professor. I can't help this. I always have to do a setup. The way that the NIH decides, we get 100,000 proposals a year, right? Scientific proposals where the scientists of the country give us their best ideas about how to make people healthier, we can fund somewhere between 8,000 and 10,000 of them, right? So it's a massive undertaking to decide which of those 8,000 to 10,000 of the 100,000 we're going to fund. There's a scientific review panel of peer reviewers. I used to serve as one when I was a professor, where it's thousands and thousands of scientists from across the country who agree to read these proposals and rate them. Now, I've mentioned earlier that one of the major problems we've had is that early career researchers have a lot of trouble getting their ideas funded. It's also true that if you come from a sort of outside of the top 20 universities, you're going to have a lot of trouble. I've had researchers come and tell me that when they moved from a sort of less prestigious university to a more prestigious university, the same proposal that they put that was rejected from the less prestigious university, literally word for word almost, is accepted when they go to the more prestigious university, right? And so you have this sort of institutional bias, I don't know if the word bias is the right word, against newer ideas, against great ideas coming from sort of less traditional universities. Stanford, Harvard, we were very lucky. But if you go outside of that, you get but there are great ideas from everywhere. So we changed how the review process works. Still the hard soul of how we decide how to which and what to fund. But we changed it so that there used to be 5 criteria. There'd be like how significant is the grant proposal? How innovative is it? Are the methods any good? Is the investigator any good? And is the institution, the university where they're at, working any good? This is for the extramural program. That procedure, I can tell you as a grant reviewer, would almost always end up focusing on will the thing work, and then on the underlying it is the investigator any good? Innovative ideas had almost no chance, or much less chance than they ought, I should say. So we've changed the grant review mechanism to focus on 3 questions: how innovative and important are the ideas? Is it possible that the thing might work? And are the investigator and environment, do they have the sufficient resources that they need to achieve it? That's now a binary, not a Nobel Prize winner gets 100% and poor fringe epidemiologist today gets 4% or something. Instead, you have a 1-0. Do you have the knowledge and resources you need to do the project? the reviews have been now focused on the most important things about those proposals. And the chance that early career researchers have, that new ideas have, is much more. And I've gotten lots of feedback from my friends who now are serving as reviewers, telling me this is way better discussion, way better, more in-depth understanding of the ideas and more fair evaluation of the ideas. It's been actually heartening, because I was convinced that everyone was going to freak out, because that procedure for evaluating scientific projects has not changed in decades.
Yeah, so just to add to something Jay said earlier, so, Jay deals a lot with the outside. My job mainly is dealing with the inside of NIH, the people, working at NIH. And it's been interesting, Jay sort of alluded to this earlier, to experience various meetings when we've made various changes and have people say, gee, we've been talking about this for years and you did it in one meeting. And so for, another example that kind of tags on to what Jay is talking about, when I was acting director, I met with the, at the time, director of our Center for Scientific Review, and she came to me and she said, scientific review should be centralized, and she explained to me how it had currently been working. And I immediately said, well, of course, this makes complete sense. So just to explain, as Jay was talking about, all our grant applications go through scientific review. Now, when I came in as acting director, we had a Center for Scientific Review, which did about 70% of that review, but still 30% was being done in the institutes by their own reviewers.
It's like grading your own homework, right? They have to grade their own homework.
And so she actually showed me examples where there had been sort of undue influence over the review to try to direct grants in a certain direction. So it was clear in 1 minute that having an agnostic center that solely focuses on review, that has no interest in the scientists or who gets the funding and doesn't make those decisions, is the right place for review to happen. And so I immediately agreed that we should centralize scientific review, and we worked on that, and by the time Jay got in, it was well underway, and then Jay obviously agreed, and we got it done. And so now 100% of our review is done within the Center for Scientific Review, which is, again, completely agnostic. And so, but this isn't something I came up with on my own. This is an idea that had existed for years. Everybody knew it was the right thing to do. It just wasn't getting done. And within one meeting, we were able to move that forward. And so getting the feedback from the internal staff to say, hey, we're so happy you're making things move forward, to me has been very gratifying. And I've seen this across in multiple areas, both inside NIH and even within HHS, talking about our progress.
Nicole, can you talk about the Standard Organoid Modeling Center? And you and Tony are like experts in this. So.
Sure, yeah, I'd love to. I will add one tiny quick detail to the example that Jay and Matt were just talking about, which is we have this amazing director of extramural research, the NIH Deputy Director of Extramural Research, John Lorsch, and he has put into place an enormous reduction in administrative burden. So really simplifying the paperwork process for external applicants to put in, their grant proposals. Things like standardizing the biosketch form and, reducing sort of the need for unnecessary things like letters of intent. And so that's been, I think, another element of kind of the grant process. So the Standardized Organoid Modeling Center, the SOM Center, is a really cool flagship effort that is a demonstration of how the institutes and centers are now really productively working together at the NIH. So it's a partnership between the NCI, NIAID,
Nicole, tell them what an organoid is.
Okay, an organoid is basically like a miniature human tissue. So you can take a collection of cells, of human cells, induced pluripotent stem cells. So you can basically take something like a skin biopsy from a patient and take them backwards down kind of a developmental lineage, and then redifferentiate them or grow them down a lineage to make them into like a miniature heart or a miniature brain or a miniature lung. And obviously they're not going to have consciousness or have full functionality of the tissue, but it's actually remarkable the degree of biological relevance that you can achieve when you're using these three-dimensional human cell-based models. And historically, they've been used more in the area of precision oncology, which Tony is an absolute world-leading expert in, but also in things like toxicology and environmental chemical assessments. But increasingly, we are basically building out the infrastructure to allow them to be much better standardized and characterized and validated so that they can be used in things like novel therapeutic development and drug discovery. We're working very closely with the FDA, who really is prioritizing advancing these human-based innovative technologies for drug safety and efficacy, so they'll be validated for use in regulatory decisions. So it's really the type of infrastructure that the scientific community needs to advance those technologies that can really only be provided by an agency with the resources like the NIH.
So one of the things about that standard organoid modeling is that in many cases it replaces animal use in research. Sometimes you can use these cells are really cool. Like I got to visit a lab where you can see these little tiny cell things, the organoid cell, that's the technical word, things. And it's beating. It's literally beating. And then you can do studies on it to see what causes it to, sort of environmental exposure that actually damages your heart without having to do an animal model or a human model.
Yeah, I just want to say how we're taking some of these ideas to the NCI. They talk about not wanting to damage the heart, and that's of course, we need to make sure our drugs don't do that, but my goal is often different, where we do want to damage this cell that we're studying. We want to kill it. And I remember I went down this road a long time ago when I was just starting cancer research. I think my wife told me, I forgot who it was, but it was my mother-in-law who asked me, so what is it that you do in the lab? Do you just put drugs on cancer cells and see if it kills them? And my response as a young molecular biologist is, oh no, it's much more complicated than that. We need to learn about pathways and genes and axes and crosstalk. But that went away. She's a sensible lady, and I thought to myself, I've got to remember why don't we do more of that? And then later on I got involved in a project where we needed to figure out this drug, how it worked and what kind of cancers it worked on. And so I just did that. I took the drug and I put it on patient tumor cells, not cell lines, not animal models, just took patient tumor cells out of a patient, put the drug on it, and found that it killed this kind of cancer, but not this other kind of cancer. And that eventually became a very successful program with a drug called venetoclax in chronic lymphocytic leukemia and acute myelogenous leukemia. But it all started with just a very simple idea, which it's hard to explain why this is wrong, okay? If you want to know if this drug is going to work on this tumor, maybe put this drug on this tumor and see what happens. So there's a bit of technology behind that, but we're bringing that to the National Cancer Institute. And I want to actually set up a place. My vision is eventually to set up a place where people can, if they have a tumor, especially without standard of care options, this is a place that we can maybe give them a readout of, these drugs might likely work and these drugs might not likely work. And we can also do discovery with patients' actual samples rather than relying on models so much.
And after we launched the standardized organoid modeling center, I started getting emails. I know it was really strange because usually I'm getting unhappy emails. They were happy emails. Jay, thank you for putting this on. And I was like, okay, I met Nicole, I met Tony. They're like, this is a good idea, and we just did it. And now I don't know what to do with myself. Happy emails coming to me. So it's a crazy thing.
Going on what Dr. Letai mentioned about complexity, the COVID crisis really made a huge dent in the trust between professional science and the public, but this is a trend that had been growing. As research science, especially biomedical, becomes increasingly sophisticated, it becomes equally difficult for the public to conduct meaningful oversight of this science and the public funding, which is making it happen. After the sort of Doge period of last year, we saw a renewed focus on some of the stuff that NIH had been funding that was more political, ideologically driven, DEI, that sort of stuff, which I think harmed that trust even more. So NIH is in an interesting position because you sit in between the professional science and the general public. So going forward, how do you think you can try and heal this divide between professionalized science and the public interest?
I think we have to make America healthy again. I mean, that's really the only way. That's really the only way. I mean, these ideas are fun. They're really interesting as scientists to grapple with. All of the bureaucratic stuff we've been talking about is frustrating but important. The ultimate way to solve that distrust is by making people making advances in science that actually do translate over to better health for people. That's it. That's the standard.
Yeah, if I could add something. So I think what you're talking about is important. And you know, I think the key is that, and we're working with our IC directors to start thinking this way. Our institute directors, yeah, I should say, sorry, using acronyms, sorry. Is that, you know, we need to make sure that the science that we're funding and working on is actually aimed at addressing problems that people in the public really have. And the way that I think about this as a physician and a scientist is you'll hear the term out of NIH and other places, they say this bench to bedside. But I don't think that way. I think of bedside to bench to bedside. In other words, we need to look and talk to our patients, understand the problems our patients are having, understand what their needs are, what they want out of their treatments, what do they want out of their medical care. Then go back to the lab and say, how can we solve these problems? And then translate that back to the patients in the hospital, right? So this is how I think we're trying to think about it. Part of that is promoting for more physician scientists. I think we need more people who can bridge that gap and do what I'm talking about. Because, you know, a scientist in the lab doesn't know what's going on in the clinic, and a doctor in the clinic doesn't know what's going on in the lab. We need those people to make those connections. So this is something that I know I have as a big agenda item that I nudge Jay on all the time, but I know he agrees with me.
Yeah, I agree with that. But I'd say I'd add one thing on top of that, because it's not just how are you treated in the hospital or by your doctor. It's how do we live a healthy lifestyle? Like I'll give you, you know, like the dietary guidelines. Yeah, you eat real food. That seems like a good idea, right? But there's real science underneath that, and the NIH participated very heavily in making sure that the dietary guidelines, as they were released, were actually grounded in excellent replicable science. A lot of the problem then is not just what happens in the hospital, but how do you keep people out of the hospital? How do you keep people away from the doctor? We need to do all of that. The bench, amazing molecular biological advances, the bedside, how do you better manage people in clinics, and how do you better inform doctors, but also how do you better help people stay healthy?
I just want to say a little something about, sorry, I interrupted an applause line. I just, with regard to oversight, I just want to hopefully engender a little more confidence now, and that is that I've been very impressed at the way that, and I only know my institute, the National Cancer Institute, that the way the staff takes direction. So under the previous administration, this wasn't a staff-driven approach. It was driven from the top. The administration wanted, for instance, different sort of political initiatives. DEI is an example that Jay gave, prioritized across our funding program. Okay, so they did that. They were instructed, and in a way, it's appropriate in administrative structure in a government that you sort of do what you're told. But I would say that really almost by the time I started, and I only started in September, they had been told the opposite. So we want to depoliticize and get this out of the grants, and get this is no longer a priority. And essentially it was done. You should be assured that, like, this isn't something where there is kind of an indigenous movement to undermine all this again. And if so, I haven't seen it. People there are very driven by cancer science. Frankly, a lot of them could make more money working other places. They work at the NCI because they see it as a unique place to do what they want to do. And what they want to do is not get in the way of improving cancer treatment. They want to make it better. They're dedicated to that. And I just want to make the point that I do think that priority has reasserted itself pretty strongly.
Yeah, I'd love to build off of what Tony just said. Totally agree with that. And it's been really, really heartening to see the response across the NIH to something like, for example, one of the activities that predated the establishing the SOM Center was back in April. Jay released a statement on the NIH prioritizing human-based science. So the Human-Based Science Initiative, which was really dovetails with this emphasis on innovation, on really advancing the technologies that we're using to improve human health and our understanding of disease. And the reaction from across the NIH was overwhelming. It was incredible. I was getting all kinds of unsolicited happy emails myself saying, hey, we're so excited about this. We want to be part of it. Can we come detail into DBKIPSI? Can we contribute? You know, we've wanted to do this innovative work for such a long time and haven't felt this level of support from senior leadership before. So I think there's a lot of excitement throughout the scientists at NIH.
Let me confirm that. I was just telling Jay. So I was an extramural researcher up until September 29th, 2025. Okay. And coming to the NCI wasn't even the picture for me until late summer. It all happened very rapidly. But after Jay's announcement, this is like what I'd been doing with my life for a couple decades. I immediately started I got to get a hold of Jay Bhattacharya directly. And it's not so easy as an extramural researcher to just get them on your Rolodex. But now I actually have pretty good contact with them. But that was one of the reasons I was very excited to come to the NCI, was because I thought, this is great. They're going to be welcoming these ideas now, and this will be a great time to do it.
So how do we ensure that all this funding actually delivers meaningful benefits to the public? I know it's a difficult balance. We don't want to rush things to market too quickly and have commercialization be the only reason that research is occurring. But we also don't want endless streams of government funding that just don't go anywhere. So that's it's a perennial issue in science, and I'm just curious to see if what your approach might be to that.
I mean, you've heard kind of the excitement about some of the changes that we've made internally. The upshot will be the scientists of this country will focus on things that actually matter for better health for people. Let me just talk a little bit about something that Tony alluded to about DEI. The problem is that it was an ideological agenda that actually never translated over to better health for minorities. We're absolutely committed to making sure that minority populations have better health. That's part and parcel of what the NIH is about, right? We can't make America healthy again unless every single American is healthier. So the problem with DEI isn't that it focused attention on the worst health of minority populations. That's a real fact. The problem with DEI was that it actually wasn't addressing that problem. It was trying to solve a different ideological problem using the tools of science to do it. Depoliticizing then is a way of refocusing the energy of the NIH on the real problems that Americans have. Actually, in a sense, like a lot of the studies would say, well, look, you have lack of health equity, meaning that minority populations have worse health than non-minority populations. And as if the goal was to just get minority populations to have the same health as non-minority populations. That sets the bar too low. We want better health for everybody, and if minority populations end up having better health than non-minority populations, that's fine too. If everybody's health gets better, the NIH is a successful organization. Removing, you guys, I'm not used to applause lines, so you can probably tell I'm not a politician. So it's the students never clapped for me. It's just a strange thing, but yeah, I think the thing is like by removing the politicization of science and giving scientists the green light to go forward with their amazing new ideas, as long as they're focused on really making improvements in health for people, that's the way we do what you're asking.
I'll say, I think Jay may have soft-pedaled another approach I think he is interested in, and I am too, and that is, so it's actually a really tricky question. How do we know we're funding the most effective research? So here's the problem. It's a real problem to identify when a grant is put in front of you, whether that is going to be an effective grant at the end of the 5-year term, which is typical. But what's surprising is when you think about it, even after that 5-year term, it can be difficult to understand whether that grant is more effective than that grant than the other one, because to go from idea to actual, say, FDA approval can take much longer than the term of the grant. So that's a challenging project, but I think Jay will agree. We're starting to try to identify what are the features of grants that makes them a successful grant, and then what are the features that those grants have at the time of application that can help us predict success. But actually, I'm saying that because NCI is interested in that, but Jay is actually setting up an office, at least the name might be changing, but the Office of Metascience.
That's the name, Office of Metascience.
Okay.
There's tremendous controversy over this. There are people on this stage that want a different name, but they want an acronym, you guys. You're not going to be very surprised.
So that explanation makes so much sense, and my mind, whenever I hear something like that, goes to, well, why wasn't this the case before? You have various experience inside and outside NIH. Why do you think these ideas hadn't been implemented already?
Matt alluded to this. The leadership of the NIH, the direction of the leadership of the NIH really hasn't fundamentally changed in decades. But the NIH, one of the side effects of the NIH is that it creates a scientific aristocracy, right? It creates a scientific aristocracy by giving the same people the grants over and over again, often because they have great ideas, but sometimes it's not just because they have great ideas, because they have a great track record, even though their ideas are not necessarily the best right now. To win an NIH grant is a huge thing in biomedicine, in biomedical research. Right, I got tenure at Stanford University in part because I was successful at getting NIH money. And so you have an entrenched aristocracy created by the NIH. You have a whole ecosystem where people are so used to thinking, okay, only these people have the good ideas, that when you change it and you start to elevate people with different ideas, the entire establishment, as one, screams bloody murder, saying, "Oh my God, you're killing science." When in fact, what you're actually doing is you're refreshing science with new ideas that would not ever have seen the light of day, but are potentially very, very promising. You heard some of them already over here. And that's the goal. That's the goal of the replication initiative. That's the goal of the initiative to try to to identify using scientific methods actually to identify promising new ideas that would otherwise never have seen support. The goal is not to have a portfolio where every single paper, every single proposal produces 5 papers. I don't care a bit about whether there's a single paper published by proposal. What I actually care is, do they produce ideas that improve health? I'm never going to get used to that, you guys. If you have a portfolio of 50 projects, 49 of them fail, and the 50th cures type 2 diabetes, in my view, Jay, that's a fantastic portfolio. And one of the things I've been really focused on is giving the leaders of the NIH the ability to take intellectual risks, because I'm not going to judge them on whether the proposals that they pick one at a time failed. I'm going to judge them as a whole whether their scientific choices and decisions actually advanced American health.
Let me just mention one of the mechanisms that we're sort of taking advantage of that leeway Jay's given us, and that is that if I go to 5 years, because typical grants of an R01 grant, which is a standard academic grant, is 5 years. There's other ones, but that's sort of a standard grant. And one of the challenges for young, new, novel, exciting, risky ideas is that often it'll start with, you lay out your aims, and often you'll have 2 or 3 specific aims. Say, I'm going to do this, and then if that works, I'll do this, and if that works, I'll do this. Now, what if it's a really good, exciting idea, but specific aim number 1 is kind of risky, which is what we want. It's exciting, it's novel, but no one's really showed this before, so I might be wrong. Okay? Now, unfortunately, all too often the response in the past has been, well, this is too risky. This is too risky. We can't fund this because if 1 is wrong, then 2 and 3 are not going to work, and we've given a 5-year grant. So our approach to that is to split this 5-year grant into a 2+3 grant. So combined with our sort of newfound ability to use more discretion at the institute level to choose grants that are meritorious, irrespective of a really strict line in the sand pain line, combining that with our ability to identify grants that seem to have these properties. Their score wasn't perfect, but the reason why it wasn't perfect is because there was something risky proposed early. We can give those people one of these 2+3 grants, and the way that works is they get 2 years of guaranteed funding to test that first idea. So if that doesn't work, well, what we'd like them to do is go out and have another exciting brand new idea, and we'll give you another 2+5 grant if it's a good idea. But if that is successful, we could do a secondary review and say, good job, here's the other 3 years. And that's a way we see of like rescuing grants that otherwise in the past would have suffered from this too risky categorization that would make them unfunded.
Got it. So on the money side, Jay, I know you have a big background in economics as well. If $50 billion is still a lot of money, even for today. And that's just a portion, obviously, of all the research funding that goes to biomedical research every year. The healthcare and biomedical sector is kind of unique economically in that the worse the results get, the more we spend on it. And this negative feedback spiral has gotten so bad to the point where we're about a quarter or so of GDP just on healthcare in the United States. So how do you see the NIH's role in breaking this spiral? How can it try to redirect funding to also try and break up the very increasing centralization of universities, hospitals, research centers all over the country that we're seeing just grow enormously and really have a huge influence in how the research is done in the U.S.?
In sometime around 2010, 2011, the NIH made a decision that almost no press at all, but it almost ruined my career. It made a decision that it was no longer going to fund health economics research. And in 2015, it formalized that with a guide notice that says it was restricting its investments in health economics. Now, the field of health economics is a lot. There's lots of things that it does, and some good, some bad. But one of the things it's supposed to do is help answer that question. Right? Is help answer how do you I mentioned earlier about the $3 million cure for sickle cell anemia. Right? Well, if I can invest, if a research project idea comes in that says, well, we're going to find a new way to produce these cells that's at 1/100th the cost potentially, the old NIH might say, well, that's not necessarily such an innovative thing. We already can do this cure. Why not focus on other areas? In the new setting, the new NIH, I'm going to bring health economics back, and we'll fund that research that reduces the cost of care. The bias in technological change in biomedicine in the last decades, 40 years, has been toward producing amazing advances in knowledge that are so expensive that almost nobody can really get access to it. Those are the biggest success stories. The bias should be toward advances that are accessible to all the people, like for everybody. And health economics is one way to sort of refocus the NIH toward that. The idea isn't to fill the NIH with health economics. That's never going to happen. But there's already one at the top, and it's not probably too many. But the idea is to have that kind of thinking embedded in the NIH, so that people can make decisions about what projects to fund, with an eye toward not just the latest advance that's going to get you published in Science or Nature or something, but an advance that lowers the price of care, that makes it available for everybody. I'll give you a concrete example of this. There are a lot of medications that are already approved that are basically off-label or off-patent, basically free. We're investing in seeing if those medications can be used for other uses. I'll give you one that's really promising. There was an old vaccine called Zostavax for shingles. Anyone in the room take this thing? I took it. The shingles vaccine is called Shingrix. The old one was called Zostavax. It's got pulled off the market because it didn't really prevent shingles all that well. Whatever. But it turns out that in some really solid observational studies that some health economists have done and others, that it might reduce the progress toward dementia by 20 or 30%. a simple vaccine that's really basically very, very cheap. And my colleague at Stanford who told me about this, he told me he'd had a lot of trouble getting support for a randomized trial to check whether this actually works. That's a great investment. What if we actually do that and it actually works in a randomized trial setting? That means you can prevent 20 to 30% of all Alzheimer's disease cases. A tremendous health economic effect, a tremendous effect on reducing the suffering of so many people and families with a simple repurposed drug. That's the kind of ideas that I would love to see at the NIH.
Yeah, the other thing that we're trying to do as well is assure that we're spreading our research funding around, right? It shouldn't all be centralized in a few big institutions, but we have different programs for this, the Idea States program, we have other programs where we try to help new or smaller institutions that don't maybe have the infrastructure for gaining grants to be able to participate more in our grants process. And so we're working on that and really trying to get, sort of more diversity in the number of and institutions and places that we're funding.
I mean, there's great ideas all across the country, and so this is a really, really important part of this. A lot of the problems during the pandemic was scientific groupthink. And having a concentrated group of places where we make most of our investments, the Stanfords and Harvards I love the institutions, but they're not but they're not the only places where good ideas are. But by having investments all across the country where scientists are, you actually combat scientific groupthink, right? You have schools of thought that compete with each other. That's the old way, I think.
Actually make science better that way.
Let's make science great again.
One additional aspect that I'll add to that is not only are we attempting to, or successfully kind of spreading the wealth around in terms of our extramural funding, we're also, I think, spreading our reach into the communities that suffer the highest degree of chronic disease burden, so rural populations across America, and actually putting and advancing programs that are already in place to deliver best-in-class, both standard of care, but also clinical research to populations that don't normally have access to things like clinical trials. So we have a program under Poughkeepsie called Care for Health, which is one of the ways that we're going to be implementing a lot of the MAHA initiatives. And this is a network of primary care providers embedded in rural communities. And so we're leveraging new technologies, so telehealth approaches, artificial intelligence for better lung cancer screening, et cetera, to really take the best-in-class care and research advances to the communities that are suffering from chronic disease burdens in this country.
Thank you for mentioning the rural element. It's kind of ties in with what I said before, that so much of this money goes to these big centers, which tend to be urban ones. And so the participants in these trials tend to be in urban areas. So I appreciate that inclusion. The last point on that is, Jay mentioned this in his initial remarks, but our friend Jason Locasell likes to point out that for a long period of time, the average age of the NIH awardee went up by 1 year every year. So it kind of seems like it was the same age cohort of people getting the same money, and some of it will trickle down to the postdocs, right? But that hasn't changed significantly. So how do you get more ideas from the younger scientists involved so they can feel like this is something that they can actually make progress in and not just be in a postdoc and stuck in that chain forever?
So one thing that we've done that I think is going to make a big difference in this area is we've released a new strategy that we call the Unified Funding Strategy. And this has changed how our institute directors are awarding grants. So let me just take a minute to explain it a little bit. So we were talking earlier about scientific review, and the applications come in through scientific review. The scientific reviewers review it, they give it a score, and traditionally, not all of our institutes, but many of our institutes were using this score as a cutoff. So you had to be above a certain level of the score and you were funded, and if you're below the level, you weren't funded. And it was just sort of an automatic thing that they were doing. Now the problem with this is that some of the metrics that are used in this scoring include things that you really have an advantage for if you are an older, more experienced, more successful investigator, because the reviewers can see, oh, this person has a track record, they're using methods that are proven, because the ideas are maybe a little old. They have resources, so we know they're going to be successful, so they would get this higher score, which would make them more likely to be funded. So with this new strategy, the scientific review is still very important, because what we found is the scientific review is good at binning in a large way. In other words, sort of good quality or poor quality, but it's not good at small differences. So those little differences in score that could have knocked you down below the line really doesn't mean anything. So what we've done now is we've put in this strategy that Jay has given to our institute directors that requires that they take into account the scientific review. It's a very important piece. We still want to make sure what we're funding is scientifically valid work. But now that is, they are no longer allowed to use those what we call pay lines or cutoffs. They have to take into account many other factors as to what should be funded. Things like, is this an actual priority in healthcare that's something the government should be funding? Is this something that already has a lot of funding, because maybe we don't need to pour money into that. Maybe we need to go somewhere where there isn't enough funding. Are we spreading money around to people with new ideas? Are these new innovative ideas? Because they might get a lower score because the methods aren't proven, because they're new. How could they be? But hey, this is something that's high risk, high reward. If we take a chance on this, it could have a huge benefit if it works out. So taking all these different things into account is going to create huge opportunities for younger people with new ideas, I think, to get their grants funded. So I think we're going to see the benefit of this strategy in a year or two as this gets further down the line. So that's one way we're definitely addressing this.
I mean, I think that the career plight of young investigators is just, it's shocking, actually. So like you have you earn a PhD, you're a young, bright kid, and you you're great in high school, you study math all the time, and then you go to college and you major in biology, and then you do some work in a lab, and you have some great ideas, you go try to get a PhD, and most of the time is spent working on someone else's ideas. You do get the PhD, then you have to do postdoc 1, postdoc 2, postdoc 3, essentially working on other people's ideas, usually people who had large grants from the NIH that are older. And then if you're lucky, you might get assistant professor job, and then you have to fight for an NIH grant in order to get your idea. You're in your 40s or 50s before you try any new ideas out. That pipeline timeline of what it takes to become a young investigator absolutely has to change. It's the fundamental reason why there's sort of stagnation in biomedical sciences, or the relative stagnation in biomedical sciences. It's this idea that Matt is talking about will help address that by empowering young scientists, empowering our institute directors to give young scientists a chance on ideas that are really promising. That's really fundamentally what they need. Not special set-aside programs, but really a chance to try their ideas out. There was a scientist in Cambridge named Max Perutz, who was he spent a decade in the 40s and 50s in University of Cambridge trying to figure out the structure of myoglobin. All his professors told him, this is too hard, don't do this, there's not enough good technology available. He just ignored them. And somehow for a decade, he putzed around Cambridge and figured it out. And then he figured out the structure of hemoglobin, absolutely transformed hematology, he transformed medicine. The question that keeps me up at night is, are scientific institutions places where people like Max Perutz could succeed? A young investigator with a really great idea, would we give them the chance to succeed like that? And the answer right now is no. But the kind of changes that Matt is talking about sounds geeky, but it's actually fundamental. You have to give people the chance, young people the chance to try their new ideas out. And they won't always succeed, and we have to change the culture of science so it's a little more like Silicon Valley. You give a young entrepreneur a chance to found a new company, it fails. If it's a productive failure, they get a second chance. We need to bring that spirit into biomedicine.
I'll just say briefly from the NCI perspective, one of my daughters is one of these young scientists, so the matter is kept directly in front of my attention a lot.
Thanks.
Okay.
You know, I mean, I was just going to add that the other thing that we need to do, and Jay and I have talked about this a lot and are going to work on, is it's not only the scientists we have in the pipeline who already exist, we actually need to encourage our American students to go into science. This is extraordinarily important. We've imported a lot of our postdocs for many, many years. We really need more Americans to want to be interested in science, and I think something that the scientific institutions, the medical institutions haven't done well is communicating with younger generations about why it's interesting, important, and a great career to be in science. And some of it maybe we have to change the paradigms of how we set things up, but you know, there's limitations to what we can do at NIH about this, but it is something that we think about and I think is really important. We want to encourage young people to get into science here in the United States.
Amen.
And the last question from me before we get to the audience once. We probably have some Hill staff in the room with us today. What sort of stuff can Congress do to help push some of these reforms forward and hopefully, if necessary, codify them into the statute?
Well, one of the key problems, I think, in making sure that all the institutions around the country that have great scientists and great ideas have a fair chance at NIH funding is the way that we fund the facilities support. There's huge controversy over this earlier in the administration with an idea to cap indirect funding, indirect costs. People in the room, some people in the room are know all about this, but again, a geeky thing, but it actually has huge consequences. The way that we fund the facilities of the country, in order to win the facility support, you have to have amazing scientists who can win the NIH grants, right? Less than 10% of people of the grants applications we get are funded. But in order to, and the facility support is directly tied to winning the grant. If you can't win the grant, the institution you live in won't get the money to build up their facilities. But at the same time, in order to attract great scientists to your institution, you have to have great facilities. It's kind of a catch-22. It's a cycle that guarantees the rich. Walter, hi. The rich will get richer. And it guarantees that the funding will be concentrated. It would be wonderful to see a solution from Congress coming where the idea would be we separately compete for the facility support based on a market basis, on costs, right? So if University of Oklahoma can provide a square foot of lab space more inexpensively than one of the top institutions of the country, then they would have a leg up in winning the facility support. Rather than fight over what the right indirect cost rate is, let the market decide it. Let competition decide it. And that will then lead to the facilities around the country being lifted up, with scientists around the country being lifted up. And let scientists go to where the great labs, where the great facilities are.
And I'll say that we have a small exemplar of how that could work already. So we have an Office of Research Infrastructure Programs within DPKMC that has a substantial budget, a couple $100 million, and they have construction grants, they have infrastructure grants, they have shared instrumentation grants. So if a university wants a new mass spectrometer or some kind of really nice advanced technology that's very expensive, they can apply for these grants. And in line with the emphasis on innovation and on really actually supporting novel technologies in that infrastructure space, ORIP just published a new strategic plan just a few weeks ago on the website, and I encourage you to go read it. It's really fantastic. And there will be funding opportunities that will be announced in the coming weeks for this infrastructure that really emphasize innovative technologies and direct funding of that infrastructure rather than having it come out of those research grants.
And the key thing is competition, right? Allow the great institutions of this country to compete on a level playing field, not the sort of biased playing field where you have to have great scientists in order to get facility support.
All right, now for some audience questions. We'll try to get through these as fast as we can. High-level policy is good, but peer review determines funding and remains broken. How are we improving peer review?
Okay, so we've done a lot on peer review, and Matt's described some of it. The key thing is that we are focusing peer review on 3 questions, and one of which is are the investigator and environment, the institution, can they actually do the work there, right? That's it. But that's a very simple question, 1-0. It's not, do you have a Nobel Prize? We're no longer asking that question. But then the other two are, is it an innovative idea that's worth doing? And are the methods good? And then we're encouraging the institute directors to take into account all of that. In the past, what would happen is that they would take into account, will the idea work? And what Matt said is exactly all what happened. Like if you propose an idea that kind of already has worked, you're going to have a leg up in winning that grant. That's gone. That's gone on the new peer review. The institutes now have the green light to take intellectual risks that they otherwise wouldn't have been able to do under the new peer review system.
I'll also say that we're expanding out the reviewer pools as well. So we're trying to bring in, again, this dovetails with the last question on an emphasis on supporting young investigators. So we're trying to bring new blood into these reviewer panels and bring in folks that may be at earlier stages in their career, but also better able to recognize high risk, really exciting, really innovative ideas when they're doing those proposal reviews. We're also bringing in expertise in new approach methodologies and alternatives and innovative technologies. So that's another way that we're trying to
Actually, did you know, like in the old NIH, this is the old NIH, new NIH, right? So the old NIH, there was a time where in order to be a standing member of these peer review committees, you had to have an active large grant. That means you had to be well established in your career in order to be reviewing grants for other people.
It's just like the definition of an echo chamber.
Exactly, couple. That philosophy is gone.
I know one of the institutes, NIH, is alternative and holistic medicine, something like that. So the question here, will the NIH incorporate holistic medication to combat chronic disease?
So we have an institute called the, oh my God, I'm going to mess up the acronym, NICCIH, and Nicole's about to tell you what that is. National Institute of Complementary and Integrated Medicine. Thank God I have people up this can help me with their acronyms. The point is that we are absolutely interested in understanding these technologies, which is really what they are. These methods of treatment. And for too long they've been sort of marginalized as if we know for a fact it can't possibly work. When you have large numbers of people giving you testimony that it might work, the missing piece is rigorous evaluation. Taking these ideas honestly and seriously, sorting between things that actually work and don't work. And that's exactly what the NICCIH, oh my God, I'm doing the acronyms, does. It's actually quite exciting that we have this capacity because it's the way that we can essentially take these ideas. And my mom, by the way, is a big fan of them. She would yell at me all the time when I was in med school over this. And treat them seriously the way that they deserve to be treated.
We're also advancing. We're also advancing concepts like food as medicine. So the Office of I got a call too now. Hey, it's contagious. So the Office of Nutrition Research inside of Poughkeepsie is coordinating advancements in nutrition research across the agency and the institutes and centers, and partnering with a number of them to really fund ideas like food as medicine and to build out things like nutrition education programs as part of training in medical schools and residencies and nursing education.
All right, can you tell us a bit more about how the funding cuts are not being reported correctly, that it's more of a reimagining?
What funding cuts? I mean, it's been.
Let me just give one statistic. We at the NCI and the NIH as a whole spent more on extramural research in fiscal year 2025 than any year previously. You can go with that, Jay.
Yeah, the other thing to remember is if money is taken away from a program that's not performing well, that money goes to another program. So the money didn't just disappear. It doesn't evaporate, right? It goes to other science. So when they say science is being cut, science has not been cut. We spent all of our money last year on science.
And the Senate and the House, I think, are just about to pass bills that fund us this year at a higher rate, at a higher level than last year. I mean, I think I've never really fully felt like in the core of my soul what fake news is until I started reading about how we're cutting everything at the NIH. I mean, it's a political ploy in order to resist the change that we're bringing.
Matter. This one I think is for Nicole. Are the stem cells being used for organoid development ethically sourced, i.e. umbilical or adult versus embryon?
I mean, short answer is yes, absolutely. And this has been in the news recently because of the announcement, I think it was last week, right, about prohibiting the use of fetal tissue in research. But that's something that the NIH has eradicated actually already. I mean, so the stem cell work that we do is in induced pluripotent stem cells. So that was the sort of scientific explanation that I gave a few minutes ago about how you can take a patient's own cells and then turn them into stem cells and then use those to do things like grow organoids.
All right, let's see here. Last week, it was reported that several NIH review panels are at risk of losing all members by the end of 2026. How is the NIH working to ensure that the advisory councils are fully operational?
Yeah, so that's so let me explain for 1 minute the council system. So we have these advisory councils that people are appointed to and then rotate off. So this is a normal process. People come on, they come off. It's a constant process, and as they come off, new people have to be appointed. And sometimes that process takes time. There's a lot of conflict of interest, ethics that has to be dealt with. Finding people who are willing to do it and put in the time. It's a process, and this has always been a challenge of NIH across all administrations. In fact, when I came in as the acting director and I met with our committee management folks, the slates for the councils dating back 3 years hadn't been seated yet. So this is always something that's been behind. So actually, we have taken steps to make this move much faster. And actually, I've been working with our institute directors to get a larger list of possible candidates and move them through the process faster so that we will fill up these councils quickly and have the appropriate members on these councils to do the work that they need to do to help us with our funding. So I think you're going to see over the course of the next year, this problem is essentially going to be solved.
And actually, can I just add on to that? One of the things I've asked the institute directors to do is to find younger researchers to be members of these councils, right? I think that if you want these councils, these are, by the way, the peer review that we've been talking about, that happens, that's not what the council is about. The peer review is filled out. That's not been a problem. If the councils sit at the sort of the last step in saying, okay, yeah, the institute directors have made good choices about which of the grant proposals that come in from the outside should be funded. I want the young, early career, promising researchers to sit on those councils as well as the people with gray hair like me. And so I've asked the institute directors to make sure that younger, early career researchers have a chance at these councils. That was started by an early career researcher, I'm sure. So.
How can we ensure that any progress made isn't undone by changing political leadership?
You can get paralyzed with this. Like, oh, if I make a change, well, maybe it'll get undone. The key thing, I hope you've heard this, none of these changes are politically motivated. Not a single one of them. What they are motivated by is actually making science work and make science work for better health for people. If a future political leader wants to reverse that, God help them.
Right. The answer is make it work. Make it work, and it'll be hard to change.
Can you explain the difference between NIH and ARPA-H?
Yes. You can use. I actually love ARPA-H. So although maybe I'm a minority on the stage, no, probably not. I'm just kidding. Okay, so ARPA-H is an agency that was created, I think, 3 or 4 years ago. The idea actually was because the idea was that the NIH was failing to identify innovative research. It's about $1 billion agency. And so the task that ARPA-H has been given is to identify big, high-risk, high-reward ideas and fund them. And I applaud any agency that's doing that, and especially the ARPA-H under the Alicia Jackson's leadership is aimed at improving, using those big high-risk, high-reward ideas to improve health. And so, God bless them. I think that's a really, really good thing. But to me, it shouldn't be an excuse to make it so that the NIH isn't doing the same thing, right? The ARPA-H in part exists because the NIH of the past failed to do that. And now we have two agencies that are committed to that same goal, to take big risks with our ideas and translate them to better health for people.
And that is, I think, evidenced by the fact that the more innovative programs that we have launched at the NIH have actually attracted attention from ARPA-H. So ARPA-H has come to us and said, hey, wow, this complement animal research and experimentation complementary, this common fund program is really fantastic, and we want to kind of jump on board, and we want to give you some money towards that. The SOM Center, they're really interested in partnering on the SOM Center. So there's a high degree of coordination between NIH and ARPA-H right now.
And just, Jay keeps pointing out that I like to tell you the geeky stuff. Just to clarify something that might be confusing, ARPA-H has always lived in this kind of limbo. Is it part of NIH? Is it not part of NIH? And you'll see it in our budget, for example, when you look at budgets when they come out. So it's in this weird limbo where it is sort of lumped in with NIH, but it does function independently under HHS. So we don't direct ARPA-H, but we collaborate very much with ARPA-H.
Yeah, and the nice thing is that it's actually good to have two different takes on how to invest in innovative research. I think like people ask me about ARPA-H, I think it's a good idea. They actually, when the legislation that led to it, some of the materials around it actually cited work that I published in support of it. So, I like it. It's good to have these two takes on the same goal.
Well, Dr. Memoli, Dr. Bhattacharya, Dr. Letai, and Dr. Kleinstreuer, thank you for your candor and your courage. Let's give it up for your new NIH leadership.